Welcome again to our chigger learning videos. This time we will discuss about chigger mite collection and global morphological identification. Chigger mites are mostly found in suburban or rural areas. In these areas, humans live close to gardens, vegetation, fields, or natural water sources. Chigger mites live in moist and slightly alkaline soil, covered with plants and vegetation, with a high humidity of about 80% and shaded areas with temperature of approximately 25 degrees Celsius. Chigger mites' habitats include low bushes, plantations, around water sources, garden and vegetation, riverbanks, forests, and rice fields. Black plates are commonly used to collect chiggers. In the possible breeding sites, a set of 5 to 20 black plates will be laid in the desired area with 2-meter intervals, either horizontally or vertically. If the plate lies on a chigger mite breeding area, chiggers might crawl past. Their creamy to reddish color make them easy to be spotted on the black plates. Each black plate is observed for any movement during a 2 to 10-minute period. If a moving chigger is spotted on a black plate, a transfer pipette, which was previously dipped in ethanol, will be used to suck the chigger and transfer it to an ethanol-filled tube. A hand lens can be used to ensure the chigger is inside the tube. To prepare the slides for chigger identification, the content of the tube is emptied into a glass dish and observed under a stereo microscope. Using a fine bristle, individual chiggers are separated. One chigger will be picked and laid on the object glass to let ethanol dry and avoid clumping. A drop of mounting medium is placed on an object glass. Then, the separated chigger is set to a good observation position on the mounting medium. Lastly, a cover slip is placed on top. The slide is labeled and the chigger location is marked. These slides are left for approximately 1 week for the mounting medium to dry. Lacquer or nail polish can be used to seal the margins of the cover slip. This slide is now ready to be observed under a compound microscope. In our last video, we discussed about chigger mite's life cycle. This is how each stage looks like under a stereo microscope. Taxonomically, chigger mite belongs to the phylum Arthropods. Arthropods are classified into 2 big groups based on their mouthparts, those with mandibles, like insects, and those with chelicerae, like arachnida. Chigger mite belongs to class Arachnida. Adult stage of members of this class have 4 pairs of legs. Class Arachnida is divided into 3 orders: scorpions, Araneae or spiders, and Acari or mites. Chigger mite belongs to family Trombiculidae, hence are also called trombiculid mites. They are also known as berrybug, redbug, harvest mite, scrub itch mite. However, the most well-known term is chigger mites. It will only bite host on its larval stage. Like discussed in the previous video, the larval stage has 3 pairs of legs, whereas the adult stage has 4 pairs. Now, let's discuss important morphological characteristics of chigger. From the upper or dorsal side, the major body parts for identification include mouthpart or nathostome, consisting of chelicerae or pincer-like claws, palpus or supporting appendage near the mouth, and galea or hair between chelicerae and palpi. the hard dorsal plate or scutum, dorsal hair or setae, and legs, including claws and special setae. For identification purposes, we can ignore the normal setae and focus on the special setae. On the lower or ventral side, important identification points include the coxal setae, anus, and the ventral setae. More detailed identification characteristics will be discussed further in our next video. That's all for today. Don't forget to click the like button and subscribe to our channel. If you have questions, please leave a comment below. Thanks and see you in our next video.